remember, I would like to re-announce the open house at the Joseph Moore Museum on Saturday. They've got a lot of interesting station plans. It's going to be distanced and safe. They will, you know, if you're interested in science, and I know you are because you're here, it's not anatomy and physiology, but it is science, and it's interesting, and they've put together they worked very hard on putting together something that early students could enjoy doing. So that's Saturday from 10 to 4. Let's see. I have your quiz 2, which I will grade today, and I will post the answers to Moodle today, and I will send you out your midterm, I will input your midterm grades on Banner, you know, the mid-semester midterm grades will be inputted tomorrow. So the grades on Moodle will not include your lab, the contribution of your lab scores, and everybody's getting 100 on that for all practical purposes. So the total on Moodle is a little bit lower than what I will report on Banner for your midterm, because the midterm will include half of your semester lab scores. Okay? So grades are going to be available soon. I've already input the grades on Moodle for your problem set, and after class today I will make my answers available so you can look at your answers and look at my answers, see what you got perfect and where you probably lost points. And I'm happy to go through it with you. I didn't mark up your papers because it's electronic, but I have my marks so I know where I took points off. So that's the past. The future is a problem set 3 due on Friday. Again, open book, open notes, work together. Turn it in on Moodle Friday by 9 p.m. is the deadline. Again, the problem set is trying to get you to work with some of the anatomy and physiology to help you understand it. And then quiz 3 is due on Sunday. Quiz 3 will quiz you on the anatomy of sensory systems. No muscles, no brain, sensory systems. So eye anatomy, ear anatomy that we're going to cover today, somatosensory anatomy that we covered last week, some olfactory or taste anatomy that we'll cover today. So anatomy related to sensory systems is on the quiz. So you can focus your studying for that. My goal for this rapid bam, bam, bam of problem sets and quiz is to help you prepare for exam 2, which is coming up on Wednesday. The material that you will get into your brain for the problem set and get into your brain for the quiz will help you get it and keep it in your brain for the exam on Wednesday. Happy to answer questions at any time about focusing your efforts, what to learn, what not to learn. Brittany's available for tutoring, both drop-in on Tuesday, which is the day before the exam, or email her if you want to schedule something outside of the drop-in. Okay, let's get to work. Today this first part of the class is going to be on how we hear, the auditory system, how we convert sound waves, which are a pressure wave in the air, into something that our brain can interpret as sound. Humans are pretty good at this. We have pretty good hearing, and not as good as bats or something, but we have pretty good hearing, and not as good as dogs, but pretty good hearing. And we are very good at detecting language. We can pick out language in a very noisy background because we're evolved to hear human sound, especially language. We won't get that psychology, we won't get into that, but we are going to try to understand how we turn pressure waves in the air into electrical signals in our brain. Okay, my first My next slide is actually a video that I want to show.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration, and sounds of lower volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth, known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spinal passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the 2 fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire.
That's wrong. We'll fix that.
The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
Okay, having woken up to Beethoven, let's unpack a lot of the information in there. So you can read on Moodle, will be this PowerPoint, it's already there. You can re-watch that if you're interested. Okay, some anatomy. Ear. The name, the formal name for the ear is the pinna. It's got some parts, the earlobe and the tragus, but let's not worry about the detailed names for the anatomy of the ear. The auditory canal, or the external auditory canal. So pressure wave moves air in the auditory canal. That pressure wave moves the eardrum, or tympanic membrane. The tympanic membrane is coupled to the auditory ossicles. There are 3 auditory ossicles: the malleus, the incus, and the stapes. You may have heard these referred to as the hammer, the anvil, and the stirrup. Looks a lot like a stirrup. I will, if on a quiz, I will accept both the Latin names and the English names. Malleus, incus, and stapes. Move. The video dwelled on ligaments and an angle, and a pivot axis. Not a key thing for me. I don't teach ligaments. But I do teach bones, and I do want you to know the names of the bones. The stapes is what moves in the oval window, moving fluid, the perilymph in the ducts of the cochlea, and it moves because there is the round window, not listed here. The round window is a place, it has a membrane there and allows the pressure of the stapes to move the fluid in the cochlea, and then the pressure wave is released, I guess, by the round window. The round window is a pressure valve, a release valve for pressure waves. If you didn't have that release valve, and this was constrained by bone, which it is, fluid is not compressible. So the stapes, but the video made a point of saying that the stapes wouldn't be able to move the fluid unless the fluid has somewhere to go. And it has somewhere to go because of the flexibility of the round window. So the oval window is the input of the vibration of the perilymph, and the round window is the output valve for the pressure wave in the fluid of the cochlea. Cochlea, the middle ear is the cavity in which the auditory ossicles are located. This is often where children and adults get ear infections. Eustachian tube is a tube that connects the middle ear to the mouth. We're not going to go into that too much. And the vestibulocochlear nerve, which has two branches, the vestibular branch, which we'll talk about in a few minutes, and the cochlear branch, which we'll talk about in a few minutes, carry information from the cochlea and from the vestibular apparatus to the brain. This is one of the cranial nerves. So this is anatomy to know. Here is a simplified view of the cochlea. So here's the eardrum, the tympanic membrane, malleus incus stapes, moving fluid, which is called perilymph. That's a name that you do want to know. That's anatomy. Perilymph in the scala vestibuli. So pressure waves go up the scala vestibuli and then return in the scala tympani. Return to the round window, which is the escape valve. In between the scala vestibuli and the scala tympani is the cochlear duct and the basilar membrane. These are going to be important for transduction in just a few seconds. You can cross off helicotrema. That's not a name that I need you to know. Yes?
Did you spell the fluid that's in the...
Perilymph, yeah. Perilymph. Perilymph is the fluid in the scala vestibuli and the scala tympani, and another fluid that we're going to talk about in a minute. So that's in the scala. And there's another fluid called endolymph, which is different, which is in the cochlear duct. I'm going to move the camera just for a minute so people online, you can't see it anymore. So I'm happy to answer questions if you have them. All right, so here's another blow-up of the eardrum, the movements of the auditory ossicles, the movements of fluid up the scala vestibuli, down the scala tympani, released by the round window. This one's not that important. This is important. So the video mentioned that sounds of different frequencies are mapped in different locations on the cochlea. And it's because of the physical properties of the basilar membrane. So the base, the input end, so this is the oval window and this is the round window. Perilymph in the scala vestibuli. Apex is a word that I do want you to know, not helicotrema. Apex, so base is the input, apex is the top. Vibrations in the oval window, vibrations in the perilymph of the scala vestibuli to the apex, and vibrations returning to in the perilymph of the scala tympani round window. Vibrations, tones, frequencies are mapped onto this structure such that low frequency sounds preferentially vibrate the apex, low frequency sounds, low frequency sounds, or vibrating your apex. Really high, I can't do it, but very high frequency sounds are preferentially vibrating the basilar membrane of the base. And it has to do with the physical properties of the basilar membrane. So high frequency waves are restricted here and vibrate here and are transduced by hair cells here. And so the nerves that carry information from here are carrying information about high frequency waves, pitch and amplitude. Low frequency waves can propagate all the way to the apex, and so the hair cells here are preferentially vibrated by low frequency waves, and so the nerves from the hair cells here are carrying information about low frequency sound. So this is a tonotopic map. Tone, frequency, pitch, is mapped on the cochlea. High frequency at the base, low frequency in the apex, and intermediate frequencies in between. So the location matters. The location of the hair cell is what is transducing sound of a specific frequency. So here's a cross section of the cochlea. Scala vestibuli, scala tympani, cochlear duct. Perilymph here, endolymph here. Here is the basilar membrane. The basilar membrane is important because it is the support for the organ of Corti. That's a name I do want you to know, because organ of Corti is the structure that does conversion. Well, organ of Corti holds the cells that do the conversion of sound waves into electrochemical signals. So I do want you to know the words on this slide, including organ of Corti. So remember, there's a pressure wave moving the fluid, and that moves the basilar membrane. The basilar membrane is the support for the hair cells. That is a word to know, because hair cells are the cells that do auditory transduction. I do want you to know the name tectorial membrane, because that tectorial membrane is movement of the hair cells, because they're supported by the basilar membrane. Movement of this relative to the tectorial membrane, which is relatively stiff, is what is going to bend the hairs, which have the hairs of the hair cells. They're not hair, they're not like this hair at all, completely different from this. But they call them hair cells, and they call it hairs. But the formal name for those structures are stereocilia. So I will expect you to use they are called hair cells, but the fibers that we're going to talk about in a minute are called stereocilia. The video suggested that hair cells fire action potentials, and that is wrong. Hair cells are short receptors, and they trigger neurotransmitter release, which activates the afferent neuron that carries information to the brain. Okay, so we're going to see that in a minute. But the video is wrong about the idea that hair cells fire action potentials. Hair cells release neurotransmitter that causes afferent neurons to fire action potentials. Is there a question? Yeah, it's very common. I have that problem myself. So I'll tell you how to make it. So the question is related to age-related hearing loss, especially at high frequencies, which is true. And I can demonstrate it for you if you want. I can play some pitch. I have a pitch generator, and I can play a frequency that will drive you crazy. Shut it off, but I can't hear it all. And my understanding of it is that it's due to death. The hair cells are fragile. And my understanding is that parts of the organ of Corti that carry information about high frequency information have been damaged by, in my case, by abuse as a teenager with loud noises like music and drugs like ibuprofen, which are damaging to hair cells. So that's my understanding about why there is age-related hearing loss. Death of hair cells. So, this is a cartoon that shows that when the basilar membrane moves and the tectorial membrane doesn't move, it bends the stereocilia. We're going to see how that turns into an electrochemical signal in just a minute. But it's mechanical, starts out, pressure wave, mechanical movement, bending of stereocilia. Here is a cartoon of a hair cell showing that it is a short receptor cell. So here's the hair cells in a support system, a support membrane. Here are the stereocilia, a bundle of stereocilia, nucleus of the cell, neurotransmitter vesicles. This is a short receptor cell, releases neurotransmitter that activate afferent nerves. We're not going to talk about the function of efferent nerves. It's complicated. So these words you don't have to know. This word you do, and the rest of these things you do. Brittany, can you dim the light so we can show some electron micrographs? So this is a cartoon of something that is just amazingly beautiful. So this is the hair cell. This is this bundle of stereocilia. Here's a bundle of stereocilia. The stereocilia have this sort of step-like pattern, which is important. So there's short stereocilia, leaning up against longer stereocilia, leaning up against longer stereocilia, leaning up against longer, until we get to one thing at the back called the kinocilia. But this structure, this stepwise structure is going to be important. And you can see that it's not artificial, it's what it really looks like. Here's this short, medium, and long. Remember that this hair cell, this particular hair cell is tuned to a specific frequency only by where it is located, on the basilar membrane. There's nothing about this particular cell that says it's a high frequency, it's detecting high frequency or low frequency, other than where it is. Everything about its transduction machinery is the same, regardless of frequency. What's different is where it is expressed along the basilar membrane. Here's another picture of hair cell bundles. Hair cells in arrays with their bundles of stereocilia. Okay, so this is a key slide because it discusses how the stereocilia convert bending into a electrical signal. So here's the stereocilia, short, long. And there's a protein, a set of proteins that are called the tip link, that link the tip of a short stereocilium to the side, the edge of the longer one. So short, long, with a connection. When the stereocilia bend towards the short, right, see, watch my fingers. When the stereocilia bends towards the short, the distance between the tip and the wall, the distance, let me do it right, distance between the tip and the wall, am I doing it? Short, long. Is that right? Am I oriented right? Okay, good. Short, long. When I'm bending from long towards short, see how my long finger edge gets closer to my short finger tip, okay? That shortening decreases the stretch on the tip link. There are stretch-activated cation channels. So decreasing stretch closes those channels. Resting potassium channels allow hyperpolarization. Calcium channels at the transmitter release site close, decreased neurotransmitter release, decreased action potential frequency in the nerve that is connected to this hair cell. So bending this way causes a decrease in action potential frequency in a specific nerve. Bending it the other way, bending it the other way increases the distance between the tip and the edge. So going from here to here increases the length, stretches the tip link, opens stretch-activated cation channels, cation influx, depolarization, voltage-gated calcium channels open, increase neurotransmitter release, and increase in action potential frequency in the nerve that is connected to it. So the bending back and forth creates a pattern of action potentials, bursts of action potentials in a specific neuron that is carrying information from this specific hair cell to the brain. So the frequency of action potentials is not carrying information about the frequency of the sound. The frequency of the action potential is carrying information about this hair cell being activated that is carrying information about the frequency of the sound. And obviously the larger sounds cause more movement, cause more neurotransmitter release, or more and less. So more intense bursts of action potentials with a higher amplitude of sound. Questions about auditory transduction. So we've turned a vibration into electrical activity in a nerve taking it to the brain. And that's sound now in the brain. There is a complicated pathway for information from the cochlear nerve, the cochlear branch of the vestibulocochlear nerve, there's a complicated pathway that includes some connection in the medulla, some connection in the midbrain, some connection in the thalamus, and then information sent to the primary auditory cortex, which is on the superior temporal gyrus. What do I want you to know about this? I do want you to know that there is processing in the medulla, in the midbrain, and in the thalamus. That is information to know. I do want you to know that the primary auditory cortex is in the superior temporal gyrus. I do want you to know that there is crossing over, but it is not as segregated as in the somatosensory and in the visual system. There is crossing over to the contralateral thalamus and to the contralateral auditory cortex. But there is some information that is staying ipsilateral. Well, I've said it, so you might as well learn it. You don't have to know about crossing over in this system. You do not have to know about crossing over in this system. I do want you to know about crossing over in the somatosensory system, touch pressure versus pain, pain temperature. I do want you to know about the differences in the crossing over in that system. I do want you to know about the crossing over in the visual system at the optic chiasm. I do not want you to know about crossing over in the auditory system. I do want you to know that the medulla, the midbrain, and the thalamus are important parts of the processing of sound. I do not want you to know the name of the nuclei. I do not want you to know the name of these nuclei. There's the primary auditory cortex in the superior temporal gyrus. The primary auditory cortex also has a tonotopic map. Low frequency sounds are anterior, high frequency sounds are posterior in the superior temporal gyrus. I do want you, just like I did want you to know the retinotopic map and the somatosensory map, that you used it in your problem set. I do want you to know that legs and torso are medial and face and arms are lateral in the somatosensory map. I do want you to know that the fovea is most posterior in the retinotopic map. And I do want you to know that high frequency sounds are mapped posterior in the auditory map of the frequency map of sound in the primary auditory cortex. Questions? We are pretty good. Okay, let me go to the next slide. We are pretty good at detecting where a sound is coming from. So not only do we detect amplitude, how loud it is, we detect frequency, how high or low pitched it is, and we detect location. We're pretty good at detecting location. So Andrew, can I ask you to help me out here? Stand in the middle of the room. Face the room. Close your eyes. He's pretty good at it. We're all pretty good at it. We can detect where a sound is coming from. The mechanisms are beautiful and complicated, and I want to teach it to you, but I am not going to test you on it. So you can just sit back and relax it for a minute, a couple minutes. I want to teach you this mechanism because it's a really interesting story, but I'm not going to test you on the mechanisms for localization of sound. We have two mechanisms for the localization of sound. One is in this horizontal plane, which was what I was testing Andrew on. The ability to detect a sound from there versus from over there versus from there. In the bottom line, sound from this position is hitting this ear a couple of microseconds before it's hitting this ear because of the speed of sound and the distance between my two ears. The speed of sound is, 700 and something miles per hour. This distance is quite a lot less than a mile. And the difference in time between the click hitting this ear versus, and then a few microseconds hitting this ear, we transduce into localization. If it hits this ear first, it's over there. If it hits this ear first, it's over there. If it hits them both at the same time, it's right there. If it's somewhere in between, it's over there. It's an amazing system. And it has to do with a coincidence detector in the medulla, the medial superior olive of the medulla. So this structure has evolved a coincidence detector, and it works like this. If the sound hits this ear first, and then it has neurons into a bunch of cells in this nucleus of the brain, it takes the action potential a little bit longer to reach this cell than it takes this cell, because this axon is a little longer than this one. And similarly, this cell, this nerve, gets to this cell sooner, the action potential from this hair cell, afferent neuron, gets to this cell a little bit sooner. So, in the middle, where they're both approximately the same length, these are the cells that are detecting coincident information coming at the exact same time, information from Brittany. If it's all the way here on the right, it's reaching, because of the delay between starting this and starting this, and this shorter path and this longer path, there's the coincidence of the two signals is not here, it's here, and these cells say, okay, must be from over there. So innervation of the cells of the medulla is what allows us to be pretty good at detecting sound in this horizontal plane. Now you might be saying, okay, Andrew, let's do another one. I'm picking on you just because you're closest. Okay, same drill. I'm going to shift my finger and point to where it is. So they're both the same in this plane, but they were different in this plane. And we can detect that difference too, but it's by a completely different mechanism. It's by the fact that our outer ear modifies the way the sound sounds on its way in. And we have learned how to interpret the spectrum of sounds that come from below us and sounds that come from above us. by the shapes of the pinna, modifying sound from this way versus the modification of sound coming this way is different from the sound coming this way. That is how we transduce location of sound. Okay, now I do want you to wake up and now we're back on stuff that could be tested. I want to transition to the vestibular system, which is also part of, it's very similar, but the cochlea is how we transduce sound, that you're now knowledgeable about, and the vestibular apparatus is how we transduce position, body position in space. And I'm not talking about proprioception. I'm talking about whether my head is like this, or like this, or whether I'm moving like this, or whether I'm moving like this, or whether I'm moving like this. All of that information, movement and movement of my head and movement of my body is transduced by the vestibular apparatus. So we're done with the cochlea for now. We're done with the cochlear branch of the vestibulocochlear nerve. And now we're getting into the vestibular apparatus. And this slide has names of structures that I do want you to know. There is a swelling of the vestibular apparatus very close to the cochlea called the saccule. So I do want you to know that the saccule is this swelling close to the cochlea. And I do want you to know that the utricle is a swelling a little further away from the cochlea. So we have the cochlea, the saccule, and the utricle. The utricle and the saccule are structured so they have hair cells in them, very similar to the hair cells in the cochlea. It's not on a basilar membrane, but it's on a membrane. They have hair cells that bend when we move. And they are tuned to linear movement. So if I move this way, or if I move this way, I'm using my utricle and saccule. It's not like one is for forward movement and the other is for lateral movement. They both work together to detect linear movement. Now you could say, well, wait a second, Bob, your muscles are moving you. You know which way you're moving because your muscles are moving you. But we could do the same thing. If I had a wheelchair or something, I could sit here with my eyes closed and somebody could slide me one way and I would know that I was sliding this way or sliding this way. It's not as easy to demonstrate as sound, but it happens. You know, even if you're not using your muscles, whether you're sliding this way or this way or this way or this way, because of the saccule, and the utricle. Hair cells in there are going to transmit movement, because movement is going to move a fluid, and the fluid is going to bend the hair cells. No, it's not going to bend the cells, it's going to bend the stereocilia of those hair cells. And that information is going to be carried to the brain by the vestibular branch of the vestibulocochlear nerve. These two nerves are going to join to form the cranial nerve, the vestibulocochlear nerve. So, saccule and utricle transduce linear movement. Fluid moving, bending the stereocilia. When I move it's basically acceleration. When I accelerate this way, when I accelerate this way, when I accelerate this way or this way, it's activating, the fluid in there is bending the stereocilia of hair cells in there, and the transduction mechanism is the same as in the cochlea. The other structures here are called the semicircular canals. They're quite circular, but this one is sort of horizontal, so you're looking at it obliquely. This one, you're looking at this way, so 1, 2, 3 semicircular canals. And these are for, and I'm not going to ask you which one is anterior, posterior, medial. Just know the name, semicircular canals, and that they are important for detecting rotation. So when I rotate my head this way, I am activating hair cells by movement of stereocilia, hair cells in this structure at the base of these semicircular canals. So the swelling is called the ampulla, and the structure that houses the hair cells is called the cupula. And so when I rotate my head, I rotate fluid, and it moves hair cells in the cupula of the semicircular canals. So I can detect whether my head is moving this way, or this way, or this way, or this way, horizontal, vertical. Three hair cells for the 3 dimensions. Three semicircular canals for the 3 dimensions. The ampulla and the cupula are structured like this. So this is the semicircular canal. Fluid moves the cupula, which bend the stereocilia of the hair cells. So you all know what it's like to get dizzy. If I do this for a few seconds, I'm not getting dizzy yet because I'm using my eyes to know where I am. But I'm getting the fluid in my semicircular canals moving. It's now moving along. And so now if I stop, I'm dizzy because the information that my eyes are telling me that I'm stationary, and the information that my ears are, my vestibular apparatus that says you're rotating that way, make me dizzy. Conflict of visual information from vestibular system information. Information from the vestibular apparatus is transmitted from the brain, to the brain, along the vestibular branch of the vestibulocochlear nerve. There is a vestibular nucleus. There are vestibular nuclei in the pons, and that work very closely with the cerebellum. As you can, since cerebellum is important for position, movement, you can imagine that this connection between the vestibular apparatus and the cerebellum is important for fine-tuning our movement. If we're out of balance, we're going to try to make a correction and not fall over. If we're running and we're stepping on something irregular, we're going to make an adjustment so that we don't fall over. So the vestibular apparatus is connected via a nucleus in the pons to the cerebellum. It's much more complicated than this, but that's what I want you to know. Vestibular apparatus, pons, cerebellum. You do not need to know the name of the nucleus of the cerebellum that is doing this. That you can cross out, and we're not going to talk about the vestibulospinal tract either. All right, let's take a good 10-minute break. That's the auditory system. That's how we hear. It's not how we understand language, but it's how you understand, that a soprano sounds pretty different from a baritone. That's how you can discriminate a soprano from a baritone. Language is complicated. We're not going to get into language. Music is complicated. We're not going to get into music. But that's how you hear pitch and amplitude and how you detect movement of your head relative to the world. Let's take a 10-minute break, and then we'll start with the olfactory system. And I want to play a little game while we're on our 10-minute break. I'm going to put in the 4 corners of this room 4 different odorants. So I'm going to open up little bottles of stuff that smells very strong. And I think the smell is strong enough to go through your mask, but if I think it's okay if you open your mask for a minute to smell it. Just don't touch it. Don't put your nose on it. Just waft it up towards you or put your nose near it. At each of the stations will be a card, and whichever station you go to, you'll pick up a card, take your pen or pencil with you, and write down the name of the odor. What is it? What is it? And then take that card with you to the next station. So you'll keep your card, and each one of these odors has a number on it. So you'll write number 3 is this, and then you'll write number 2 is this. So I'm going to put these odors in each corner. I'm going to put a card there, and then I'm going to ask you, it doesn't matter what order you go to them, just keep your spatial distancing, and over the course of the next 10 minutes, go to each of these odorant stations and identify the odor.